going to talk about basically the real analysis of a real data set using the scatter package. And before doing that, I would like to show you, again, you can actually follow me on the website, or you can follow me using the RMD file in your RStudio. So if you open file number 06, basically, this is the same information as you see on the website. But the cool thing about it is that what you can do in RStudio, you can run each code chunk using this button here. And basically, it will run it for you, so you don't need to copy-paste stuff. But make sure that you run all the chunks before the one that you want to run at the moment. So in this case, you will not get errors. OK, so what I'm going to talk about is the quality control of the expression. So here we are going to use the data set. We call it the Blischek data set, which is probably wrong now because the paper has been released on bioRxiv, and now it's a Tung data set, I would say. So before that, we called it the Blischek data set, but never mind. So this data set has data, single-cell data from 3 individuals, and for each individual, I think there are 3 replicates. And those are iPS cells. So we start with basically loading the scatter package and then loading the molecules data. It's the same data that you already used for the UMIs chapter. And also there is an annotation table which contains information about the cells in your data set. So this is basically the phenodata information that Davis mentioned previously. So then you can actually look at these 2 data sets. So that's how the molecules data set look like. So you have cells in columns, and you have genes in rows. And then these values are your expression values. So the cells have these IDs, which are explained in the annotation table. So basically, for each ID, you have information about the individual, about the replicate, about the well in which it was run, and about the batch. So now we can create a scatter object using these 2 data sets. So what we first need to do is to create the phenodata object. This object has to be an annotated data frame class. So what we do, we just call this method with this table and create the phenodata data frame. And then, as Davis mentioned before, the row names of phenodata has to be identical to the sample IDs, to the call names of your data set. And that's what we make sure here. So, and then basically the next step is creating the scatter object. So if you can run this, and if you don't see any errors, we can continue. Did you manage to do that? OK, cool. So now you have the scatter object, and then now you can actually do all the things that were explained in the previous chapter. So, but the purpose of this chapter is to do the quality control. So first, we want to do some filtering. So we will remove the genes that are not expressed in any cell. So basically, by counting the expressed values in each gene, we can remove those. And then we can also, we would like to define the controls for our data set. So these controls can be either ERCCs. So ERCCs are provided in this data set. That's why we can use them. So basically, we just find them using this function. And also, this data set contains mitochondrial genes. Those genes were provided by the authors. So we just define this list of genes which are mitochondrial. So looking at the mitochondrial genes is also useful. So if you have a lot of reads in those genes, it means that something wrong happened with your cell. So once these things are defined, then we can run this method on the scatter object. It's called calculate QC matrix. And we, in this method, we define our controls, RCCs and mitochondrial genes. So this method will basically calculate different features of your data set based on these controls automatically.
What would increase mitochondrial rates be indicative of bad lysis or?
Well, I think that one thing can be that the cell is dying already.
Okay, so it's maybe
Something like this, yeah. Yeah, so the question was what happens with the cell when the mitochondrial expression increases. So one explanation can be that the cell starts to die and you get a lot of reads in the mitochondrial genes. So it's a good control for the single cell data sets. And now we are ready to look at the data and to find out which cells are outliers and which genes are outliers. So we first start with looking at the total counts. So total counts are molecules detected by sample, per sample, sorry. And so we can plot this histogram, which shows you the number of total counts per cell on the x-axis. And we can see that this distribution is not normal, and whether actually this is one of the questions whether it should be normal or not. But we can also see that there are a few cells here which have very low number of total counts. And usually those cells are bad cells, cell outliers, so we'd like to remove them. So we define this threshold here of 25,000 total counts. And we'd like to remove those cells from our data set. You can actually choose your own threshold if you don't like this one. But that's a good idea, basically, to remove very lowly expressed cells in your data set.
Would that then depend on setting the threshold, depend on how many genes you would expect to see in total, depending upon the cell type you're looking at? So
Yeah, so the question is, does this threshold depend on the data, on your data, basically, right? On the sequencing depths or something. Yes. So for this sequencing, well, I mean, here we look at the distribution, so we can kind of see that those cells are on the lower tail of this distribution. If you have higher sequencing depths, everything will be shifted, and yeah, you will have to define threshold manually. It's not. It's hard to do it in an automatic way. So here we have the exercises. Exercise 1 is asking you how many cells does our filter remove and what distribution do you expect to see here. So again, you can find the answers to this exercise in the RMD file if you don't want to spend too much time on it. But basically, if you run this filter, if you look at the RMD file, so we found that we have those 46 cells which we think are outliers, and we'd like to keep the rest in our data set. So we create this cell filter, which is called yeah, so here we call it filter by express features. Oh no, sorry, this is exercise 2. Exercise 1. This is called filter by total counts. So this is the answer to exercise 1. So we keep this filter in our R session, and we move on now. So the second thing we can look at is the number of detected genes per cell. So we can plot a similar histogram here. And again, we can see there is a heavy tail. So So those cells basically have the number of expressed genes, which is quite low compared to the rest of the cells. So it's usually also a good idea to remove those cells from your analysis, because those are usually outliers. Again, the distribution, ideally the distribution should be normal, as in the previous case, but it's not usually the case with the real data set. So again, you have to look at your data carefully and find out what's the good threshold. So this filter is called filter by expression features in your RMD file. And again, we want to keep it in the session. And this filter, as you can see, with this filter, we remove another 120 cells from our analysis. Maybe that's not another, maybe that's... So I think we still use the full data set. So this filter removes 120 cells. The previous filter removed a different number of cells, but here we use the same full data set. So we haven't filtered those first cells yet. So these first two steps are one of the most important ones, I think. Then we can look at the ERCCs and mitochondrial genes, so basically your controls. For this, we can use the method which is called plot phenodata. And so in this phenodata, we can provide this string in text. So basically, all these features are explained in the Skater vignette if you want to have a closer look. So here we just use them. So we'd like to plot phenodata based on total features and percentage of counts in your controls in mitochondrial genes, right? And we would like to color this by batch. And this is the plot that Skater provides for you. So we think it's very short and easy way to make a nice plot of your data set. And that's why we're using Skater, actually, because it's very convenient. So what we can see here is that, so each point is a cell. And on the x-axis, we have percentage of counts in the mitochondrial genes. So we can see that some cells have quite a high percent of reads in the mitochondrial genes. We probably can remove those cells, cut off somewhere at 13%, or either we can keep them because it doesn't look that they are very strong outliers. In a similar way, we can plot ERCCs. So again, instead of mitochondrial genes, now we use ERCCs. And here, interestingly, that we can see that one of the batches, the orange one, corresponding to this individual, is definitely an outlier. So this is a first signal for you that something went wrong in this batch. So it's definitely a good idea to remove this batch before going further, or trying to think what happened wrong and trying to correct for it, and then look at the same plot again and see whether the correction worked. But definitely you have to do something with this batch. So what we do is we remove it because we think it's a bad batch. So this is actually exercise 3. We need to create filters for removing this bad batch. And also, actually, yes, we remove cells with mitochondrial genes where the percentage is more than 10. So again, you can have a look at the RMD file and do the exercise yourself. And these are the numbers that we have. So the ERCC filter removes 96 cells, and the mitochondrial filter removes 31 cells. So I'll give you some time to do the exercise.